Hello everyone, welcome to another video from DNA Bioscience. Today we are going to learn about the TCR, which is called the T cell receptor. As the name itself indicates that it is a receptor which is present in the surface of the T cell. As we have already discussed the T cell receptor in type 4 hypersensitivity reaction, the antigen presenting cell which first engulf the antigen and it chew off it and crush it. Then the antigen presenting cell it represent the antigen by the help of MSC complex in the surface. And then the T cell sense it and the TCR receptor binds to it and it activates the T cell. The function of TCR is mainly activation of the T cell. So let's discuss the introduction. The T cells are a subset of lymphocytes that play a large role in the immune response. They have specific receptors on the surface which are generally complex of integral proteins which interacts with an antigenic peptide by MSC class 1 or class 2. If it is a MSC class 1, then it is for cytotoxic T cell and the glycoprotein which is present is CD8. And if it is a class 2 protein, then it is for T helper cell and the glycoprotein which is present in CD4 on the antigen presenting cell which leads to activation of the T cell. So let's discuss about the characteristics of the T cell receptor. It is a heterodimer consisting of alpha chain and beta chain. As you can see in this picture, this is the alpha chain and this is the beta chain. It consists of two heavy chains and two light chains. We all know that. The upper part is called the F heavy part and the lower part is called the Fc part. The F heavy part is generally responsible for the antigen binding site, and the disulfide bond links the light chain and heavy chain and the heavy chain with the heavy chain. This is the hinge region, and the light chain as well as heavy chain they consist of some variable regions and some constant region. So BL is for variable region of the light chain and CL for the constant region of the light chain. Likewise, VH is the variable region for the heavy chain and the constant region of the heavy chains are CH1, CH2 and CH3. The CH2 part is composed of some carbohydrates. It helps in complement fixation. So if you just take this part, only this part, the FAB part, and place it over a T cell, then it becomes the T cell receptor. The only difference is this is one heavy chain and one light chain. In this case, there is the alpha chain and the beta chain. So the next point is only one antigen binding site is present. In this case, there is two antigen binding site. This is one and this is another. But in this case, only one antigen binding is present. The next part is TCR structure is very similar to FAB part of immunoglobulin. We have already discussed that this part of the TCR structure is very similar to the FAB part of the immunoglobulin. The next point is the transmembrane region is present to anchor us. This is the transmembrane region which is present to anchor us. Next point is CDR12 interact and combine with the MSC complex. So let's discuss in detail about the antigen binding site. In this area is particularly divided into three regions, two peripheral regions and one particular antigen binding site. So the two peripheral regions are called CDR12 and the antigen binding site is this area. So the peripheral regions, the function is they interact the MSC and they combine with the MSC. So the actual antigen binding site is the middle one and the peripheral sites they interact with the MSC and then bind with the MSC complex. The next point is a very short intracytoplasmic tail is present and the alpha chain and beta chain are linked with the CD3 chains. What are CD3 chains? CD3 chains are involved in the signal transduction pathway. So this is all about the T cell receptor. Let's summarize this once again. The T cell receptor structure is very similar to the FAB part. Only the difference is it is called alpha chain and beta chain. And the next part is it consists of a cytoplasmic tail. It consists of CD3 chains which helps in signal transduction. The antigen binding site is divided into three parts, two peripheral regions and one particular antigen binding region. I will make a separate video for CTR. So that's all for today. I hope it will help you. Thank you.